Hello everyone, I'm Kenshin. Welcome to Talking History. Today's historical hoax is the century scam of cardiac regeneration research. Please subscribe, like, comment, and share, and don't forget to turn on the notification bell. Thank you. At the end of July 2022, another paper scandal rocked the world, with an impact comparable to an earthquake. This was the publication by the renowned academic journal, Science, on July 21, titled, Academic Stain, which directly pointed out that one of the most significant papers in the field of Alzheimer's disease was riddled with a large amount of suspiciously fabricated images. The paper in question was authored by Sylvain Lesné, a neuroscientist at the University of Minnesota, Twin Cities. Published in the top-tier academic journal, Nature, in 2006, it claimed that the main cause of Alzheimer's disease is the abundant deposition of a protein called a beta*56 amyloid in the brain. Since then, a vast number of Alzheimer's disease researchers worldwide have focused their studies in this direction. Over the past 16 years, it has accumulated over 2,000 citations, and Science even claimed that the National Institutes of Health in the United States allocated over $200 million this year alone to fund research related to this hypothesis. This highly influential paper has laid the foundation for dementia research, but if it is proven to be based on falsified data, all these research achievements may come to naught. This reminds Changson of a larger-scale paper scam 4 years ago, adult stem cell cardiac regeneration. First, let's introduce the faker, Piero Anversa, who is now 84 years old. He once held prestigious positions at the New York Medical College, including Professor of Pathology and Medicine, Director of the Cardiovascular Research Institute, and Vice Dean of the Medical School, making him a respected figure in the American medical community. The story began in 2001 when he and his research team published a study in the renowned academic journal, Nature, claiming that cardiac stem cells with c-KIT-positive characteristics could self-renew and differentiate to repair heart muscles, overturning the previously held belief in the biomedical community that heart muscle damage was irreversible. However, over the nearly 10 years following the publication of this paper, no other medical research team succeeded in replicating their findings. For instance, Dr. Irving Weissman, co-director of the Stanford Institute for Stem Cell Biology and Regenerative Medicine, and his research team repeated the experiments and stated, the marrow cells injected into the heart remain marrow cells. Similarly, Dr. Charles Murray from the University of Washington in Seattle also failed to produce results and questioned the accuracy and rigor of Anversa's research. Of course, Anversa responded disdainfully to external questioning, suggesting that it was others' incompetence, implying that they were simply not skilled enough. Meanwhile, within Anversa's team, there were whistleblowers who disclosed instances of data manipulation during the experiments. One of them was Italian molecular biologist Mario Ricciardi. After being hired as a researcher in 2011, he quickly became suspicious of Anversa's groundbreaking findings, suspecting them to be fraudulent. Another whistleblower, biologist Nathan Tucker, stated that he participated in the research for only 2 months and discovered that the images and data in the paper were likely fabricated. He even recounted personally searching for c-KIT features distinguishing adult stem cells in the research samples but finding nothing. However, the next day, other researchers suddenly produced a large amount of c-KIT evidence. Whistleblowers further pointed out that the team maintained a strict level of confidentiality to punish those who questioned the research hypotheses and results. Coupled with high-pressure information control, researchers were coerced into producing experimental data that aligned with the hypotheses. Fast forward to 2012, when 8 researchers raised concerns about research misconduct at Brigham and Women's Hospital. Despite the research being under scrutiny, the National Institutes of Health, NIH, continued to fund related cardiac regeneration research with nearly $250 million, approximately 7.5 billion new Taiwan dollars, after the U.S. government launched an investigation in March 2013. The situation came to a head in 2013 when Harvard Medical School and Brigham and Women's Hospital announced a review of his papers. The following year, at the request of Harvard Medical School, the prestigious cardiovascular medical journal, Circulation, did not retract a paper published in 2012 despite allegations of data fabrication. Even in 2015, despite the closure of the lab, ongoing investigations persisted. Moreover, Anversa's oversight of several federally funded research projects attracted further attention from the U.S. government. The story doesn't end there. The National Institutes of Health, NIH, in the United States sued Anversa in 2017 for academic misconduct, alleging that he obtained project funding through falsified data and images. Brigham and Women's Hospital was ordered to pay $10 million to the U.S. government. On October 15, 2018, the well-known life sciences website STAT released a statement revealing that Harvard University had requested the retraction of 31 cardiac stem cell papers by former Harvard Medical School professor and renowned cardiac regeneration expert, Anversa. He was implicated in the falsification scandal, along with Brigham and Women's Hospital, one of Harvard's primary teaching hospitals and one of the world's leading biomedical research institutions. How serious were the retractions this time? Apart from being one of the top 20 scientists in the world with the most retractions, it caused a significant setback in related research fields and wasted a considerable amount of academic resources. The reason being, almost all related studies worldwide were based on his foundational research for subsequent exploration. Although Harvard stated in its announcement, "We will adhere to the strictest ethical standards and academic norms, and any academic misconduct that comes to our attention will be reviewed according to relevant procedures and regulations," questions arose as to why it took 10 years to discover and address this issue, but Harvard did not comment on this. Jeffrey Malkenton, a professor at the Howard Hughes Medical Institute, told the New York Times that it may be because past experts were too timid and afraid to take a stand against authority. The original whistleblower, Mario Ricciardi, was passionate about the research because he himself suffered from cystic fibrosis, a lung disease characterized by fibrosis. If stem cells could repair human tissues, there was hope for treatment for the disease. However, he later underwent a lung transplant to prolong his life. The treatment turned out to be a lung transplant to extend his life. In fact, even in the fields of genetic editing, stem cell regeneration, and other biological sciences, which are considered star fields of 21st century science, there is no shortage of large-scale academic misconduct scandals. Examples include South Korea's Hwang Woo-suk and Japan's Haruko Obokata. Now, how did this scandal come to light? Ironically, it wasn't an internal revelation. According to Science, the whole incident originated from two investors who wanted to profit massively by shorting the stock of Cassava Sciences, a company developing drugs for Alzheimer's disease. They believed that if they could expose flaws in the drug's research process, it would lead to a collapse in the stock price. As a result, they paid $18,000 to Dr. Matthew Schrag, a neuroscientist and internist at Vanderbilt University, for an investigation. Shortly after, Schrag began examining the pharmacology behind the company's drug and eventually discovered suspicious aspects of the papers written by Sylvain Lesné. In key data charts, there was almost 100% similarity in multiple instances representing protein accumulation, meaning two different mice yielded nearly identical data, which is almost impossible in nature. Further investigation revealed suspicions of data manipulation in an additional 10 papers related to amyloid proteins. Even though the key figure, Sylvain Lesné, did not come forward to clarify during the entire investigation.